Welcome to this video about reverse phase protein assays. In this video, we'll explore what reverse phase protein assays are, how it works, and how it can be used to detect disease. We'll also discuss some of the advantages of reverse phase protein assays. Reverse phase protein assays is related to microassays, so before going through this video, you'd better understand the meaning of microassays. Luckily, we've made videos related to these topics before, so you can watch these videos in our channel first. Reverse phase protein assays, also called RPPA, is an antibody-based microassay technology that performs immunoassays on thousands of samples simultaneously, including tissue or cell lysates, serum, plasma, or other bodily fluids. The most important part of RPPA is RP, reverse phase. Reverse phase refers to the fact that the analyte or antigen is immobilized as a capture molecule, rather than immobilizing an antibody as a capture molecule. Reverse phase protein assays involve immobilizing cell or tissue lysates in small spots onto solid supports, which are then probed with primary antibodies specific for proteins or post-translational modifications of interest. The primary antibodies are then detected using a secondary antibody conjugated to a detection system. The signal intensity is proportional to the amount of protein present in the sample. So how does it work? To perform an RPPA, the first step is to extract proteins from the biological sample of interest, such as cells or tissues. The sample is then denatured and diluted to ensure accurate and consistent protein concentration across different samples. The prepared protein samples are then printed onto a solid support, typically a nitrocellulose membrane or a glass slide. This is done using an arrayer, which deposits small spots of each sample onto the solid support in an ordered grid pattern. Once the samples are immobilized on a solid support, specific antibodies against the proteins of interest are added. These antibodies can be fluorescently labeled or tagged with enzymes for detection. The solid support is then incubated to allow the antibodies to bind to their respective target proteins. After the incubation period, the solid support is washed to remove any unbound antibodies. The bound antibodies are then detected using various methods depending on the labeling technique used. For fluorescent labeling, the slide is scanned to measure the fluorescent intensity at each spot. For enzyme-labeled antibodies, a substrate is added that produces a detectable signal upon enzymatic reaction, and this signal is measured. The resulting signals are quantified, and the intensity values are normalized to correct for experimental variations. The normalized data can be further analyzed using bioinformatic tools to compare protein expression levels or activation states between different samples or conditions. RPPA was first originated with the aim of capturing microscopic stages of progressing cancer lesions within individual patients. This was tested by applying RPPAs to the analysis of the state of protein signaling checkpoints involved in regulation of pro-survival and growth regulation during the transition from histologically normal epithelium to prostate intraepithelial neoplasia, and then to invasive carcinoma. The new technology provided direct quantitative evidence that suppression of apoptosis via increase of phosphorylated AKT and concomitant decrease in activated ERK in human premalignant and invasive prostate cancer may contribute to cancer cell survival by shifting the ratio of cell death to cell survival. As a targeted proteomic platform, RPPA is complementary to mass spectrometry profiling and is capable of quantifying relative levels of total protein expression as well as post-translational modifications, such as phosphorylation as a measure of protein activity. RPPA was initially designed for identification of alterations in protein signaling pathways in cancer, but is easily scalable for assessment of protein pathways in other diseases and cell biology processes. Relative quantitative comparisons of protein concentrations can be made within or between multiple case studies. Antibody-antigen interactions are highly dependent on the relative abundance of the antigen-antibody species, as well as the binding affinities between the probe antibodies and the immobilized antigens. Consequently, protein samples are arrayed in miniature dilution curves to ensure that the analyte of interest remains within the linear range of detection. Such internal calibration is not possible for tissue fragment arrays or antibody arrays. Similar to tissue arrays, multiple patients are represented on the array format, so that a molecular hypothesis can be tested on a series of cases to achieve statistical validity. This technology has been developed into applications. For example, TheraLink Technologies used reverse phase protein array assays to develop a panel for breast cancer. It measures phosphorylated proteins in tumor tissues to check the active targets for drugs. Thank you for watching.